Human skeletal muscle progressively declines as we age. Healthy, dense fiber bundles slowly atrophy, while stiff connective tissue builds up in the empty spaces. To track this deterioration, researchers examine thin cross-sections of muscle tissue stained with hematoxylin and eosin. This standard dye method allows pathologists to manually observe structural changes under a microscope. But zooming in on the muscle fiber's control center, the nucleus, reveals a conflict in scientific literature. For decades, studies have disagreed on exactly what happens to the myonuclear domain as we age, with contradictory reports whether these nuclei shrink, swell, or disappear entirely. This contradiction exists because traditional histology relies on manual human evaluation. It is highly labor-intensive, subject to individual observer bias, and struggles to quantify complex tissue architecture consistently across hundreds of samples. To isolate the actual mechanisms of tissue aging, pathology must transition away from a qualitative art and become a high-throughput, objective science. A 2026 study published in Aging Cell addressed this problem by applying an automated AI pipeline to the genotype tissue expression, or GTEx, database. By running the algorithm over a massive library of whole slide images, the researchers removed human subjectivity. This image displays how AI translates biological matter into mathematical data. It isolates cellular structures, applying strict bounding boxes around nuclei, while stripping away surrounding noise. Processing thousands of these instances uncovered a definitive hallmark of aging muscle: progressive nuclear enlargement. We need to deconstruct the exact architecture of this algorithm, analyze the specific distributions it extracted, and examine how these physical distortions map directly to molecular decay. AI-driven histology transforms visual complexity into standardized data, exposing structural shifts in skeletal muscle that are missed during traditional pathology reviews. The researchers deployed this analysis across 974 distinct histological whole slide images. This dataset spanned muscle tissue from human donors ranging from their 20s through their 70s. Because whole slide images contain billions of pixels, they are first tiled and fragmented into computationally manageable patches. These patches are then fed into a neural network paired with a mask RCNN model, specifically Detectron-2, trained strictly from nuclear instance segmentation. Here we see the model's precision at high magnification. It accurately detects and isolates individual nuclei, ignoring the dense muscular fibers and surrounding connective tissue matrix. Isolating the nucleus is critical because it acts as the cell's genetic control center. Morphological distortion here is a known indicator of cellular stress and senescence. By systematically stripping away the noisy biological background and extracting the nuclei across thousands of slides, the researchers engineered a clean, high-dimensional dataset. With the nuclei accurately segmented, the deep learning pipeline extracted specific morphological measurements, primarily their exact diameter, and organized this data across decades of life. This histogram illustrates the resulting size distributions, categorizing nuclear diameters into intervals. Notice the healthy baseline in younger samples, indicated by the white bars. The overwhelming majority of young nuclei are tightly clustered in the small 2 to 8 micrometer range. As donor age increases into older cohorts, shown by the dark gray bars, the distribution drastically skews to the right, filling the 12 to 18 micrometer range. This population-level data demonstrates that tissue aging is not characterized by random decay, but by a highly specific, directional physical swelling of the nucleus. We can quantify this skew directly to confirm it represents a genuine biological trend, rather than a visual artifact. This violin plot tracks mean nuclear diameter against continuous age progression. The upward slope of the red regression line mathematically solidifies enlargement as a significant hallmark of muscle aging. But we must address a counter-hypothesis. Could the overall number of nuclei in the tissue, rather than their individual size, be the primary driver of this age-related change? This second chart measures nuclear counts. While the number of nuclei does slightly increase with age, the nearly flat regression line indicates the effect size is negligible when compared to the shift in their diameter. As a fail-safe, the researchers needed to ensure the AI wasn't mistaking age-related SAT deposits for enlarged nuclei. They manually measured carefully selected, artifact-free tissue zones, confirming the automated model's accuracy matched rigorous human review. Nuclear size, far outstripping nuclear quantity, stands as the definitive morphological metric of aging skeletal muscle. This raises a critical biological question. Is an enlarged nucleus just an inert structural balloon, or does this physical swelling reflect a breakdown in the cell's genetic programming? To answer this, researchers utilized gene set enrichment analysis. By taking the histology slides and matching them directly back to the exact RNA sequencing data from those same donors, they could effectively peer inside the measured nuclei. They isolated the genetic profiles of the 20 donors with the largest nuclei and compared them directly against the 20 donors with the smallest nuclei. The genetic profile of the small nuclei aligned with healthy protective programs, displaying high activity in DNA repair and anti-inflammatory functions. In contrast, the large nuclei transcriptomes were dominated by cellular decay, enriched for chromatin remodeling, mitochondrial dysfunction, and cellular senescence. Morphological size serves as a direct mirror for molecular reality. An enlarged nucleus is a genetically senescent nucleus. If nuclear enlargement truly represents biological decay, it should appear in diseases that accelerate tissue breakdown, independent of the patient's chronological age. For the final validation, researchers examined muscle biopsies from pediatric patients suffering from inflammatory myopathies, conditions characterized by chronic muscle inflammation and degeneration. Despite being children, these patients exhibited the exact same severe nuclear enlargement observed in the elderly cohorts from the GTEx database. This phenomenon, defined as premature histological aging, proves that chronic inflammation drives this structural shift, regardless of the calendar year. By uniting deep learning with large-scale tissue data, researchers resolved a historical paradox and successfully built a scalable muscle aging clock. Nuclear morphometry is no longer a subjective guess. It is a quantified tissue-level biomarker that integrates physical architecture with molecular decline.